Right, so microbes are constantly facing environmental changes, and so they have to be able to adapt very rapidly to be able to survive. Adaptation is what allows organisms to fit into their environment better and be able to reproduce. Some organisms, some microbes, have the ability to send signals outside of a cell and receive information from outside back in the cell so that they can kind of monitor what's going on. Other organisms can sense when there are multiple cells in a population near them. So there's more cells. They can actually turn on or turn off certain genes depending on the number of cells in the environment. Adaptation is the ability, so adaptations occur on the genetic level, true adaptations do, and they allow organisms to survive in environments better than other organisms. Natural selection is basically the survival and reproduction of the most well-adapted organisms. And so this picture is kind of goofy because obviously beavers don't have chainsaw hands, but this would be awesome if a beaver were born and had a little chainsaw for a hand and he no longer has to use his teeth to chop down the trees to make their beaver dams. And so he's all upset. And so he's like, oh, maybe if I try hard. Adaptation does not involve trying. You don't have to try to be able to survive in an environment. Adaptations are on the genetic level. You either will or you won't survive. And they occur via mutations within your genes. Some organisms have evolved the ability to alter genes on the surface of their cell. And so I use Trypanosoma brucei, these little tiny, really beautiful looking blue protozoans. These little suckers live inside of your blood, or not in your blood cells, but in the blood plasma, and they cause all kinds of problems. Because they have the ability to change or alter their surface antigens. For those of you who've taken anatomy and physiology, you remember that an antigen is what elicits an immune response, and surface antigens are what identify the cell type. So if it's a cell in your body, the antigens are called self-antigens. And if it's a cell that's not from your body, the antigens are going to be foreign. And your immune system will respond to those foreign antigens. The problem with antigenic variation is that as soon as the immune system gets activated and goes out to attack, the proteins on the surface of the protozoan change. And so the immune system doesn't recognize that specific unique antigen, and so will not attack. And then by the time a new immune system attack gets ready to go out, because you have another immune system cell that recognizes the foreign antigen, says, oh my gosh, this is bad, makes an army, the protozoan has changed its surface antigens again. So this allows the pathogens to basically evade the host's immune system and can actually cause problems for years and years. Neisseria gonorrhea can do the same thing. The one difference between Neisseria and Trypanosoma is that Trypanosoma are eukaryotic, whereas Neisseria are prokaryotic. And so we can target their ribosomes, which don't change with the protozoans. It's hard to target any specific thing since they are eukaryotes and we are eukaryotes. So many genes are expressed on what we call operons. And an operon is a group of genes together that include an operator region where the control of the gene is going to be located, a promoter region where the RNA polymerase will bind, and then the genes itself. And so here's what an operon might look like. Here you have the promoter and operator site, and then over here you have the actual genes that are going to be found in the messenger RNA molecule. A lot of our genes are called constitutive genes. Constitutive genes are the genes that are always being produced, so we don't really have, I mean, we have control over them, but the control is a little bit different there because we always need the enzymes for the glycolytic pathway. It's our oldest, most common pathway for energy production. Whereas we only have to turn genes on for, say, lactose metabolism if lactose is in the environment. So those type of genes would be called inducible genes. We have them turned off unless we need to turn them on. And then a repressible gene would be one that we have turned on typically unless we have enough of the end product. And so this is typically how we control amino acid synthesis. Those are called repressible enzymes, always turned on unless we need to stop them. We can control the production of our proteins using either positive regulation or negative regulation. Negative regulation uses repressor proteins to block translation or transcription from occurring. And that repressor protein will bind to a region in the control portion of the DNA called the operator site. And when you have a repressor protein bound, then RNA polymerase is not going to be able to move along the RNA molecule. You can have two types of repressor proteins. You can have repressor proteins that are always bound to the operator site until an inducer comes along. And when the inducer comes along and binds to that allosteric region of the repressor protein is going to come off of the DNA molecule, off of that operator region, and then we're going to transcribe the structural genes. You can also have a repressor protein that never binds to the RNA or to the DNA molecule unless a co-repressor, typically whatever the end product of that enzymatic pathway, that will end up being the co-repressor. It'll bind to the repressor protein, and together they will bind to the operator site, and then DNA polymerase, RNA polymerase won't be able to produce the transcript. So here we see RNA polymerase bound, but there's a repressor protein bound to the operator site. RNA polymerase cannot produce that transcript. Here we have an inducer that's bound to the repressor protein, so the repressor protein can no longer bind to the operator site, and so RNA polymerase will produce the transcript. Down here in repression, RNA polymerase binds and the repressor protein can't bind, so RNA polymerase is going to produce the transcript. But when a co-repressor binds to the repressor protein, the repressor co-repressor binds to the operator site, and now RNA polymerase cannot produce that transcript. So one of the most, before I talk about the most famous operons, I'll talk about the positive regulation. Positive regulation uses activators. Activators enhance protein synthesis. So they bind at a region next to the promoter site, and when they bind, they allow RNA polymerase to bind more easily to the promoter region. Most activators have to have some type of an inducer that allows them to bind. The activator I usually talk about is the cap protein, and cap protein needs cyclic AMP to bind for the cap protein to be able to bind. So now let's talk about one of the most famous operons that most people have heard of called the Lac Operon model of gene expression. And this is found in E. coli. And so this operon is going to run only if lactose is available and glucose is not available. So if lactose is absent, then the repressor protein is going to be bound to the operator site. This is an inducible operon. And if the repressor protein is bound, then RNA polymerase cannot produce a transcript of the enzymes to break down lactose. So here's the promoter site, here's the operator site, and here are the structural genes. Here's the repressor protein, and here is the RNA polymerase. RNA polymerase can't produce a copy or an RNA form of the functional genes because the repressor protein is bound. But if lactose is available, lactose binds, and that allows the RNA polymerase to produce that copy. But the Lac Operon isn't just an inducible operon using a repressor protein as a controller. It also has positive regulation. So it has negative regulation using a repressor that is controlled by an inducer. Then it has an activator called the cap protein that needs cyclic AMP available to bind. Cyclic AMP is going to be only available when there's very little glucose present. So cyclic AMP is inversely correlated with glucose. When we have a lot of glucose, we have very little cyclic AMP. When we have very little glucose, we have more cyclic AMP. And this is important because if you have a lot of glucose, then the Lac Operon won't run, even if lactose is available, because glucose is going to be used for energy, not lactose. And so here we have, from a different textbook, but a very good picture showing if lactose and no glucose is available, what happens. So if lactose is available, but no glucose is available, then the cap binding site is going to, so this is that activator region, is going to have cap bound. RNA polymerase can bind to the promoter site, and the functional genes will be produced. Because here's that repressor that has lactose bound. If lactose is available and glucose is available, then lactose will move that cap or the protein, the repressor protein off the operator site, but since cap doesn't bind because there's no cyclic AMP, then RNA polymerase cannot bind. It needs the cap protein to bind so that RNA polymerase can bind. So we won't produce any of the enzyme. If there's neither lactose nor glucose, then cap is going to be bound, so RNA polymerase can bind, but we also have the repressor protein bound, so nothing is going to be produced. And then if glucose but no lactose is bound, then you're not going to have the cap protein because there's glucose, so no cyclic AMP, so cap can't bind, and there's no lactose, so the repressor protein is bound, and RNA polymerase will not run either. All right, I'm going to stop here, and I will upload these videos and get into Chapter 8 shortly. Have a great day.